Right, Assalamualaikum and very good morning to our students. Okay, so today we learn about a little bit about proteins. Okay, so nanti kita akan masuk dalam chapter protein insyaAllah minggu ni ataupun next week. Alright, so today you will continue your laboratory number 3 or experiment number 3, test for proteins. So please be ready with your manual. Okay, and then I will show you the synopsis of the experiments. Okay. All right, so today's experiment is laboratory number 3, test for proteins. So this test is to make sure how you detect the presence of protein inside the food, inside the substance, or so forth. Okay. So before we start, the introduction for your experiments. Actually, introduction is show the synopsis of your experiment actually. So you can find the answer from that introductions. If you read properly, okay. All right, so introduction, the treatments of protein with a copper sulfate in alkaline solution is called Biuret. Okay, so the test will produce what answer will be the purple to pink violet color. Okay, pink to violet color because of presence of copper sulfate. Okay, once copper sulfate react with the substance inside the protein, you will get the pink violet color. Okay, so why? Because for any substance that contain two amine group or carboxyl groups inside the proteins, directly we join together. Together with what? Together with copper sulfate. So the free amino acid does not give the positive test. So positive test, mesti dapat warna pink or violet color. So if we get rather than other color, such as blue or red range, there is not what we call a proteins. Okay, the exact color depend on protein involvements, but it will be in the blue or red range. Okay, so we do what we call heavy metals, ions such as plumbum, mercury ions, denature protein, probably by reacting with thiol ester group. Okay, thiol groups, okay. So these thiol groups is SH and carboxyl group is COOH, okay, which contribute to the toxicity of the heavy metal ions. Okay, we use also certain acids such as trichloroacetic acids. So what happen? These things will do the complete denaturations and precipitate. Okay, what's mean of denaturation? Denaturation is a process of cutting bond of the two molecules, and then the product will be precipitate. So what's mean of precipitate? Precipitate adalah mendapan, okay. Daripada proses eksperimen tu akan dapat mendapan. So when treatment with the trichloroacetic acid is often used to stop enzyme catalase reactions. So another one, we're using ethanic and picric acids, and their derivative will also denature protein, but less effective than trichloroacetic acids. So once we put acid ataupun tannic ataupun picric dalam proteins, awak akan dapat berlaku apa yang berlaku akan berlaku proses denaturations and precipitates. So let's say eksperimen yang mudah boleh awak tengok adalah bila awak campurkan susu dengan jus limau ataupun oren. Okay. So selepas tu akan dapat macam ada particle ataupun precipitates. So that's the meaning of the complete denatured or precipitate happens with the presence of acids from oren. Alright. So the amino acid molecule contain an amino groups which is NH2, which has X base. What's meaning of base? A proton acceptor. And carboxylic acid function is COOH, which behave as an acids, a proton donor. So in the presence of acid, the amino groups is converted to NH3+. Okay. Bases will convert to carboxylic acid to carboxylate anion. Kenapa carboxylate anion? Sebab COO negatif tu. Itu dikenali sebagai carboxylate anion. The isoelectric point for the amino acid is a pH at which both functional groups are in their ionized state. Under this condition, the negative carboxylate anion, yang ni this one, right, of the one molecule exert an attractive force on the positive ammonium groups of another molecules, and a precipitate will be observed. Right? That's meaning of process isoelectric point. Okay. So we quickly go for the experiment. So objective to observe denaturation proteins by heavy metal, cation, and acids. Last one, number 2 will be determine the isoelectric point of cations. Okay. So first experiment to detect the presence of protein. Number 1, we're using a Biuret test. Kalau baca dalam introduction, apa yang ada dalam Biuret test, awak kena check . So pipette 2 ml of stock solutions of albumin into the test tube. Okay. Add 2 ml of 0.1 molar sodium hydroxide and then 5 drops of 1% copper, okay, to sulfate solutions. Okay. So apa itu Biuret test? Biuret test contain of sodium hydroxide and copper sulfate. Right. And then you have to observe the color. Okay, observation, record the observation. So this one example of another qualitative test. Okay. So changes of color. So kalau nak tahu changes of color, you have to read the introduction to the first paragraph, right? And then you have to repeat with the others proteins, such as peptone and caseins. Okay. Right, number 2, precipitate of protein by heavy metals. So heavy metals apa dia? Heavy metals, for example, adalah copper sulfate, mercuric nitrates, or lead acetate. Lead nama lain adalah plumbum. Pipette 2 ml of protein solutions, albumin, casein, and peptone in 3 sets of the 3 test tube. Each of test tube is going to be added with the 4 drops of heavy metal solution, which is copper sulfate, mercury nitrate, and lead acetates. Okay, this is the experiment, gambar aja dia, right? So you need a 3 test tube for albumin. So each of the test tube, number 1, 2, 3 here, you put only albumin. So this albumin will react the copper 2 sulfate. This albumin, number 2, tube number 2 will react with the mercury nitrates. Number 3, lead acetate. Okay. So quickly done another 3 test tube for caseins. But the solution or 4 drops adalah sama, right? And then peptone. And then fill the test tube. Again, record the observation. So dalam ni, awak kena record macam mana? So you have to record, is it precipitate or denatured happen? Is it, kalau ada present precipitate, maknanya berlaku proses denaturation. Okay. And then add another 5 drop of 0.1 molar copper sulfate, and then record the observation again. Okay. Okay, test 3, precipitate of protein by acids. Okay. So what type of acid we use? Sulfosalicylic acid, picric acid, tannic acid, and trichloroacetic acids. So this one we need about 4 tubes each of it. So you put about 2 ml of protein solution. So what type of protein solution? Albumin in 4 tubes, caseins in 4 tubes, peptone in 4 tubes. So done in the 3 sets. Okay. Why 3 sets? Because we have solution of albumin, casein, and peptone. Why 4 test tube? Because you have to put 5 drop of acidic solution each of it. Okay, so what type of acidic solution? Sulfosalicylic acid, picric acid, tannic acid, and trichloroacetic acids. Okay, as shown in the diagram, Okay, you need about 4 test tube. Okay, so each test tube we have to label 1, 2, 3, 4. So each of the test tube you have to put 2 ml of albumin. Then each of test tube number 1 you have to put the same solution. So tube number 1 will be only sulfoxylic cyclic acids. Tube number 2 picric. Tube number 3 tannix. Tube number 4 trichloroacetic acids. And then swill all the 3 sets of test tube. And then you have to record the observations. Again, is it present of precipitate or not? Is it present of precipitate? So we consider it happen on process of denaturation. So and then you put about 2 ml of 0.1 molar sodium hydroxide in each of test tube slowly and record the observation. What happened after that? Okay. So for the tips for the experiment here, if you put 0.1 molar of sodium hydroxide after this experiment, on the F you get there will be a change back into the clear solutions. Okay. So test number 4 is summarize of all test number 1. It's summarize of all test number 2 and 3. Okay. So first of all, test number 4 is isoelectric point of cations. So we need to prepare cation solutions. Okay, number 1 we dissolve in 0.1 gram or 1 pellets. So 0.1 gram itu lebih kurang 1 pellet lah untuk cation of sodium hydroxide in 250 ml of distilled or deionized water. Okay, so masukkan 0.1 gram awak punya cation pellet ke dalam 250 ml of sodium hydroxide. Okay. And then add 1 gram of pure cation powder. Okay, stir well until the solution is clear. A slightly haziness is not uncommon. So akan berlaku juga proses haziness, tapi tak common, right? So you need 2 molar of HCl, hydroxide, chlorine, acid hydrochloric. Add 4 ml of concentrated acid slowly and with stirring to 21 ml of deionized water. Okay, so 2 molar acid hydrochloric letak sebanyak 4 ml of dalam tu dan kacau. Ke dalam 21 ml of deionized water. Okay, so this one is prepared by the lab assistants. Now you have to set up the series of 10 test tube, which is labeled from 1 to 10. Okay, so kita siapkan 10 test tube, labelkan daripada 1 sampai 10. So setiap test tube tersebut, you have to get exactly pH between 3.8, 4.0, 4.2, 4.4, 4.6, 4.8, 5.0, 5.2, 5.4, 5.6 pH. So how you going to get it? By using this one. Okay. So campurkan 4.4 this one dengan 0.6 ml. So insyaAllah akan dapat 3.8. To confirm, you have to use a pH meters. Okay. So you follow this manual, you can get exact the pH on each of test tube. Okay. So bila dapat 10, and then add 1 ml of cation. Okay, macam mana? Daripada 10 test tube tersebut, awak kena letakkan 1 ml cation on each of the test tube. So we need about 10 ml of cations. Okay, swill the test tube and then record the observation after 30 minutes. Okay, so that what we call legend ataupun penanda untuk menunjukkan bahawa berlaku proses tersebut. So let's say test tube number 1, is it colorless? Is it cloudy? Is it precipitate? Kalau ada, awak nyatakan kat situ on conclusion, right? Which mean that kalau dekat jadual ni, awak letak satu lagi kolom, awak letak situ observation ataupun conclusion. Okay. Right. So from that exact you can get from here ialah cloudiness will appear by will clear up again as the acid is distributed throughout the system. Eventually, it will reach a maximum as the isolated point is achieved. The system will then begin to clear up as additional of acid continue, and the cation structure is converted to acid protonated form. Okay. Once the isolated point is reached, as indicated by the failure of the cloudiness to the fade, the continual additional of HCl or acid hydrochloric will cause the system to clear up again as the pH drop below the isolated point. So the change can be reserved by adding 2 molar sodium hydroxide dropwise. As before, the cloudy appear will persistent at the isolated point, then clear as the pH rise by the further addition of sodium hydroxides. Okay, so from this experiment, there are what we call 10 series of test tube. Daripada sini kita tengok dekat mana kedudukan isoelectric yang kita pilih. So dekat sini dia kata kalau ada keadaan isoelectric, akan berlaku proses cloudiness daripada 10 test tube tersebut, dia akan berlaku macam mana, iaitu ni, ini cloudiness to fade daripada 10 test tube tu. So kita punya tugas untuk dalam eksperimen ni, kita check dekat mana yang ada keadaan clear dan tidak ada precipitate. So bila ada keadaan clear dan precipitate tu, dia tinggal sebagai setelah reach isolated point daripada 10 test tube tadi. Okay. So hint for the experiment, isolated akan berada pada pH 4.6. Okay. So bila berlaku proses 4.6 ni, awak akan nampak kedudukan dan keadaan mana berlaku proses isolated tersebut. Okay. So untuk continue, untuk kembali kepada kasal, kita kena main dengan kehadiran HCl ataupun sodium hydroxide. Okay. So bila kita masukkan HCl, dia akan clear balik. Tapi bila kita campur dengan NaOH, semua eksperimen tersebut akan clear balik. Right. So there are questions you have to answer in this experiment. Okay. So untuk face to face, untuk ODL tak payah, awak kena jawab soalan post saja. So question number 1, describe the principle of Biuret that cause positive sample to turn purple. Number 2, explain the function of 0.1 molar of copper sulfate added in experiment of precipitate of the protein by heavy metals. Number 3, explain the function of sodium hydroxide added in the experiment of precipitate of protein by acids. Number last, discuss the event that occurred during isolated point of cations. Okay, so this one is a post question for experiment. Tapi soalan ni untuk you all jawab lah, untuk bantuan untuk jawab untuk final test nanti, okay, ataupun test 2, test 1, right? Right, is there any question sebelum kita sambung kepada the principle of experiment today?
Sir?
Yeah.
Kalau soalan yang 4 markah tu kan sir, nak jawab point ke ayat yang macam paragraf tu?
So the better 4 point lah. Dia bergantung pada soalan, jawapan. Dia bergantung pada markah.
Oh,
If we say 2 marks, you have 2 points. If we say 4 marks, 4 points lah. It's better to do like that lah. Kalau comparison, wow, buat dalam bentuk jadual. Boleh?
Okay, boleh.
Okay. Alright. So is there any question here? Left? Ada soalan lain lagi sebelum kita sambung lagi? To know the principle of the experiment? Okay, kalau tak ada kita sambung lagi. Alright. Alright, so laboratory number 3, test for proteins. Okay, notes ni saya dah masukkan dalam U-Feature. So, boleh tengok nanti, alright. Okay, what makes proteins? They are what we call 3 micronutrients. Okay, proteins, fats, and carbohydrate. In other words, we call it micromolecules. Okay, so these macronutrients or macromolecule provide calories or energies that we need in our bodies, right? So protein is an essential macronutrient in building muscle mass, commonly found in animal products. They are also present in other sources such as nuts and legumes. So itu pun ada protein juga. So protein is a basic components of living cell base being made by the carbon, hydrogen, oxygen, nitrogen. Okay. In a series of amino acid, what we call monomer, and linked by the special bond, what we call peptide bond, and form polypeptide polymer. A protein molecule can have one or more polypeptide chains. The 3 types of proteins are fibrous, globular, and membranes. Okay, so these are our example of peptide, how peptide bond was born. Formation, okay, a bond ataupun formation ataupun form. So kita ada kumpulan carboxyl here, right? And then ada one be amin, kumpulan amin. So bila gabung ni, kita akan release of the water, and then you get a peptide bond. So what is peptide bond? COONH. This is our peptide bond. Okay. Okay, we go for the Biuret test, right? So this is exactly the color you can get from the experiment. Okay. So testing protein sample with the copper sulfate in alkaline solution. Okay, kita buat Biuret test untuk apa? Untuk kita test protein kita dengan copper sulfate, tapi dalam keadaan beralkali. Okay. So positive result with the sample contain peptide bond. Okay, let's say kita ada CONH ni. Okay, bila dia bergabung dengan copper sulfate, copper ions, awak akan dapat warna ni. Okay, so the copper 2 blue present in the Biuret reagent bind to the nitrogen atom at the peptide bond and undergoes reduction to copper 1. Okay, reduce kan daripada 2 kepada 1. So that's why we get a violet color. Okay, so since this is a great test, is not greatly disturbed by the presence of amino acid in the sample, it can be used to gauge the concentration protein in the whole tissue sample. Okay, kita cuma detect kehadiran what we call amine group je. Okay, ataupun peptide bond yang terhasil. So this peptide bond akan bergabung dengan cuprum ions daripada cuprum sulfates. Okay, that's a Biuret test. So make sure in alkaline solution. Bukan acid Kalau acid kita dapat result. Okay, continue. So what's meaning of heavy metal and acid denaturation? Okay, so heavy metal is a mercury and acids. Okay, this one is experiment number 2 and 3. Okay, so heavy metal using a mercury or plumbum. Okay, acid using trichloroacetic acids. Okay, so these 2 types of substance will denature our protein. By reacting with important functional groups, they are maintaining the structure integrity of the protein. So kehadiran asid dengan heavy metal ni, dia akan mengganggu structure protein tersebut. Okay, so bila structure telah pun diganggu, okay, mungkin dia akan pergi ke arah peptide bond, mungkin dia akan pergi ke arah carbon, oksigen dan sebagainya. Akan berlaku proses what we call denatured. Okay, so what's meaning of denatured? The structure is no longer stable, therefore it become denatured. So mostly proses denatured berlaku pada 2 makromolekul. Yang pertama adalah proteins, yang kedua adalah nucleic acids. Okay, So once it becomes denatured, there will be a precipitate solution happen. So what's meaning of precipitate? Denatured and precipitate is solution turn into colloidy or cloudy. Okay. So kita ada eksperimen tadi, kalau letak oren dalam susu, akan dapat partikel denatured dan juga precipitate. Partikel-partikel putih. Right. So let's say this is our folded protein. Protein dalam bentuk folded sebab apa? Sebab dia ada banyak ikatan. So dia jadi macam ni, gumpalan ni. So bila berlaku denatured, dia akan mula unfold sebab ada ikatan yang telah diputuskan. So bila unfold ni lah kita akan dapat partikel-partikel ataupun precipitate. Right. Okay, experiment number 4. So the principle between experiment number 4. So this is the principle of isoelectric point proteins. Tapi dalam eksperimen, awak akan nampak kedudukan isoelectric itu between precipitate dan juga cloudy. Itu adalah jawapan dalam isoelectric point dalam eksperimen. Okay, so the best pH is 4.6. So 4.6 itu adalah kedudukan isoelectric point dalam proteins. Protein yang kita guna dalam eksperimen adalah cations. Okay, so what's meaning of isoelectric point? pH is that the pH in which both amine group and carboxy groups of protein are in their ionized stated. So, macam mana bentuk ionized stated tu? Amine group akan mendapat satu lagi H dan bentuk charge positif. Dan mana datang H tersebut? Daripada kumpulan karboksil. So bila berlaku sini telah mencapai isoelectric point. Okay. At this pH, the attractive force between positive and negatively charged group lead to the precipitate of the protein molecules. Okay. So dia akan jadi ada slightly precipitate dalam pH tu sebelum dia jadi betul-betul precipitate alright. So keadaan itulah berlakunya isoelectric point. Okay. So we have three keadaan okay. First cation, another one will be anions. Kalau cation in the low pH, okay, NH3, so COH akan dapat H juga. Okay, we go for PK1. And then kalau deuterion, make sure nampak kehilangan satu H tadi. So, okay. Tapi kalau anion in highly pH, NH2 kita akan kehilangan H bersama juga dengan COH. H pun tak ada. Okay. That's meaning of the anions. Right? Okay, the video will help you on understanding the practicals. Okay, so please buka semua YouTube ni, semua cerita tentang kita punya eksperimen test for proteins. Okay, ada 5 sorry, ada 6 yang boleh tengok. Okay, each of it represent the experiment yang berkaitan dengan test for protein for today. Right? Okay, ini ada soalan lama. Alright, kalau boleh jawab juga boleh tengok. Okay, boleh tengok sebab saya dah bagi dalam ni di future, boleh check juga Alright, is there a question? Sebelum saya bagi post question pada awak untuk awak jawab. So don't forget to answer your discussion question okay. So is there any question? Ada soalan tak? Sebelum saya lepaskan awak untuk menjawab soalan dan juga menjawab post question. Post question lab. Ada soalan?
Tak ada.
Soalan? Tak ada?
Tak ada.
Okay, kalau tak ada apa-apa, saya akan bagi awak punya link untuk post lab questions.